Hello, everyone. It would be hard to conjure up a title for a presentation that's more motivating than today's, entitled Preventing Alzheimer's and Small Vessel Disease in the Aging Brain. It's estimated that the continuum of Alzheimer's disease, which includes Alzheimer's disease dementia, prodromal Alzheimer's disease, and preclinical Alzheimer's disease, affects about 400, give or take, million or 22% of all people over the age of 50. That's a lot of people. And vascular dementia, which is the second most common form of dementia, accounts for about 15 to 20% of all dementias, so also very common. And in many cases, the two may be inextricably linked and contribute to one another. As far as small vessel disease, I don't think I know there's not a day that goes by if you're looking at MRI scans when we aren't reading a report that says white matter hyperintensities that are often attributed to small vessel ischemic disease. In fact, there was a registry of healthy young and middle-aged adults where white matter hyperintense lesions, or what's called periventricular hyperintensity, or enlarged perivascular spaces, were present in 2/3 of this young, otherwise healthy, young and middle-aged population. So not altogether surprising, that that's the case, given that the population prevalence of obesity and hypertension and diabetes and prediabetes and dyslipidemia is so high, and the underdiagnosis and undertreatment of these are so pervasive, and they happen to be the most prominent risk factors for small vessel disease. So to speak to us about preventing Alzheimer's disease and small vessel disease in the aging brain is a good friend, a colleague, and actually my fellowship mentor at the University of Toronto, Dr. Sandra Black. Dr. Black is a professor of medicine and neurology at the University of Toronto. She's an internationally known cognitive and stroke neurologist. She's been actively engaged in well over 70 clinical trials over the past 3 decades. She's published over 650 peer-reviewed manuscripts. She has an H-index of 132. For those of you who don't know what that is, that's extremely high, and has over 70,000 citations. In a research career that really spans over 3 decades and has bridged dementia and stroke, and has used standardized quantitative neuroimaging, cognitive, functional, and neuropsychometric measures, genetics, and neuropathology to study brain behavior relationships in the common dementias, as well as with a focus on the interrelationships between small vessel disease and neurodegeneration. So it's an honor, it's a pleasure to have Dr. Black here to teach us today about how we can think about preventing small vessel disease and Alzheimer's disease in the aging brain. Dr. Black, the floor is yours.
It's a real pleasure, David, to have the honor of presenting at one of these wonderful symposia. And I really appreciate the opportunity to talk about one of my favorite subjects, which is interactions between neurodegeneration and small vessel or vascular disease. So without further ado, I will just try to move forward. I hope I can move forward. There we go. Because I do have some consulting positions, advising that I do to some of the companies that are involved in development of drugs. I don't take any investigator fees. I've had some personal fees from the ad hoc consulting, but I mostly struggle on the underfunding that we have in Canada, David, as you may know, compared to other countries for various peer-reviewed institutions, and I do get some salary support from my fellow physicians. So my key messages today are, to jump to the bottom line, is that comorbid neurodegenerative protein and vasculopathies, proteinopathies, vasculopathies, are the rule, not the exception, in aging and the common dementias, and many now can be seen with advantages in neuroimaging and even in the blood. Multimodality positron emission tomography, where you can actually see amyloid deposits, and you can see glucose uptake, and MRI are becoming informed by genetics. We also can do CSF biomarkers and blood biomarkers, and then statistical advances are allowing us to approach precision diagnosis and potential treatments accelerated by artificial intelligence methods that use reliable, you have to have reliable ground truth, lots of validation, proper QC and validation, but that is also making this more amenable to us in the future for use in clinical care. Parallel rapid development of potential disease-modifying therapies promise mitigation and secondary prevention, but optimizing vascular health and lifestyle choices, physical exercise, diet, sleep, not being obese, social engagement, all play an essential role. So I really want to underline that because of your introductory remarks. So let's just quickly do a historical overview. This was Alzheimer, who described August Dieter, published on his findings of pathology in 1906. At age 51, she'd lost her memory, language, had delusions and hallucinations. We now know she actually had a mutation, an autosomal dominant mutation for Alzheimer's disease, discovered many years later because I think her brain ended up in Salpêtrière or what in Paris, and eventually they were able to determine that. What he described on the left side, you can see it, a normal brain. What he showed was atrophy in the back and inferior parts of the brain with sparing of the sensory motor strips. This is depicted, in the staging system that was introduced, the Brackenbach staging system. And the plaques were these little patches in the brain. Actually, they form around a venule very often, or a capillary, almost as if they're an abscess. And the tangles were indications of dying of the kind of axons that seem to be part of this disease. So let's jump through the next 120 years and just quickly run through some of the things that happened. So he described it in 1906. For 50 years, it was thought to be the disease of a rare disease of younger individuals, like she was 49, even though that was the average life expectancy at that age. And it wasn't until the 60s that the Alzheimer disease prominence was really very important. It's a whole story that I can tell you if we have time to discuss because it actually had to do with NPH and sampling of people's brains when they were in their 60s, finding out that 60s and 70-year-old people had the same plaques and tangles as this young onset condition. And this kind of began to lead to this idea that it might be much more common than was thought. So I have a little kind of local thing about the Alzheimer's Society in Canada, but I can tell you in the US, this led to the foundation of the National Institutes of Aging, and Alzheimer's disease became considered the commonest cause of dementia. So carrying on, in the 1970s and 80s, some of the molecular biology was discovered, including that there was amyloid in the plaques. And in the 90s, there was tau in the tangles. The genetic link to autosomal dominant disease was discovered. And the first drugs were starting to be used, the cholinesterase inhibitors. And then the first disease-modifying therapies, a vaccine was tried. It was a disaster. And so the vaccines were left alone for the next 2 decades, but stay tuned, they may come back in a different way. And finally, you start to get more drugs approved. This time, this was for memantine, which is, again, neuroprotective and not so useful. But the cholinesterase inhibitors were actually really important for giving people more ability to focus and maintain some sort of executive function and some ability to, kind of think, even though it wouldn't bring back memory. So the brain imaging milestones in Alzheimer's disease really reflected the development of positron emission tomography in the 1970s, actually. And it was noted early on the glucose PETs, the deoxyglucose PET scans, that there was kind of a signature for Alzheimer's disease. It was bilateral, biparietal, showing, I can show you on the next slide, biparietal and a bit on the bit frontal and also temporal. And so there was this idea that there might actually be a signature on the PET scan, but it was kind of not very precise in terms of localization. The hippocampus isn't very well seen on PET, but people were doing activation studies and receptor studies. And so the idea of imaging, like inflammation imaging, for example, Dr. Norberg, began to take root. Okay, so that was the imaging, but now let's just step back and realize the hubris of thinking we're going to solve this easily. This just shows you the systems biology approach, and you can pick whatever career you want for any of these omics and spend your whole life trying to understand it. So this is just the humility slide. That means, we focus on one little thing, amyloid and tau, and we try to remove it, or we try to do something to it in a very complex being called the human being and the human brain. So whenever I feel that we're making progress and I get over kind of optimistic, I do kind of step back and think we really are playing with fire when we just focus on one thing, but sometimes you get away with it. Likewise, the genetic landscape is much better understood now in Alzheimer's disease, and it's much more complicated than we thought, with some causative on the left side. You can see sort of causative links, and then other ones that are sort of contributory. The ApoE4 shows up because that's something that's associated with later onset Alzheimer's disease. It seems to make the brain leaky and sort of more likely to start causing trouble, maybe in your 70s and 80s, but it's not the same as young onset, where you get sick in your 20s and 30s. Shown here, on the left side, you have autosomal dominant, where you inherited 50-50 chance that you're going to get the mutation. And you can see that people get, this is showing their amyloid and their tau and the range, and it's tight. People are in bad trouble by 50. They're usually dead in their 50s, and it can start in their 20s. And that's also, being studied now with antibodies and trials going on, mostly through Washington University and the Diane-2 TU studies. So we are trying to do something for that, but it's a very aggressive disease. Then you have the ApoE4. This is the common variety, where maybe if you have one more common risk, one E4 allele from your parent, you have about 3 times the risk of developing later onset Alzheimer's. If you have 2, you have 13 times the risk. And you can see here that you're starting to lay down amyloid maybe 20 years before you actually become symptomatic, which may be in your 70s. But there's a pretty big spread there. In the ApoE4 not related, where it's just we don't understand all the factors, it's a much kind of broader, harder to get your arms around, but that's a more common reality. So about 25% of people in at least the Caucasian populations will have the E4, one of the E4 alleles. About 4% will have the double E4, maybe 21% a single one. So that just gives you a little bit of an approach, but we need to know a lot more about different ethnic groups. So with that in mind, I think you really should be thinking of Alzheimer's disease as what we may call the vascular Alzheimer's spectrum disorder. And that's because, first of all, age, that's aging of the vessels, which contribute very much to what happens in Alzheimer's disease. High blood pressure, high cholesterol, the E4 I just mentioned, diabetes, which was already mentioned, or obesity, which can lead to diabetes, atrial fibrillation, cardiac disease, previous stroke, smoking, fat, sugar, salt, poor sleep, obstructive sleep apnea, physical activity. These are all very important. And also just recently, something from a whole different area, which is hearing loss, has emerged as a risk factor for Alzheimer's disease. I'll come to that, show that later on. But this is just to remind you, you have to take care of your vasculature, your vascular health, in order to have a chance to live a life that is less likely to end up in Alzheimer's disease. Even just light physical activity. This is showing you the brain volume that they looked at in some studies that have, looked at the number of steps that people take. And it's just to show you that the more active you are, you get even more activity if you're exercising, more actively. But even just walking, and the cutoff was around, like, one study said 10,000 steps, one said 8,000 steps. People did a meta-analysis, and they said 7,000 steps every day just walking is good for your brain. Keep that in mind. So fitness and of the brain and fitness related to the heart and the vasculature, we got to hold hands together, and we've got to, and more there's more interest in the brain heart connectome, because we've made a lot of advances in cardiac disease, and we now need to make similar advances in brain health. So the twin curses. So let's start. Well, why are the vessels so important? So I just love this slide. It's freely available on the web, and it shows the brain's vascular network. I'll say more about it in a moment. And underneath that vascular network is something that you have to appreciate. We all focus on the neuron, this little fellow here, right? The child, as it were, of the family. But the family is the neurogliovascular unit. And that includes the endothelium, the actual, the vessels, the capillaries or the arterioles, the vessels that are important to provide the blood, which brings you oxygen and glucose. There then are, and it can expand or dilate or get bigger depending on where the brain needs the food, as it were. But then it's very important to know that there are protector cells. So, for example, the astrocytes, if you want to be, well, we're not going to sort of make any analogies to human beings, but the astrocytes protect the blood-brain barrier so that toxins don't get in so easily. They form these little, kind of foot processes that, prevent some substances getting in. The pericytes are very important for, they're kind of guard cells that fight off, invaders or make trouble for things that are trying to damage the brain. And then the myocytes, that is the muscle cells, help to expand or contract the spine. So you can see that all these are very important for flow regulation, immune surveillance, trophic support, and basically flow. So here's the thing that really, to me, is so amazing. They have estimated the length, I think it's of the vasculature, it said capillaries in the adult human brain, it's 400 miles. Talk about packing a lot of important stuff into a small space. The surface area is about 12 square meters between the blood and the brain interstitial fluid. The blood-brain barrier is about 0.3 to 0.5 millimeters thick. It restricts the free exchange of solutes between blood and brain in order to prevent entry of toxins. And that's partly what happens over time. And one of the things that the E4 does, it makes your brain less able to protect itself from, it makes it more leaky, less able to protect itself from those toxins. The astrocytes are in close proximity to both the neurons and the capillaries, the capillary endothelial cells, so these foot processes. And this is the key to both synchronizing neuron activity for metabolic demands, but also they regulate synaptic transmission and neuronal firing. And really, the whole neurovascular unit has got to be healthy for you to have a healthy brain. So what about this amyloid beta? There's actually, which has become the bad guy. There's sort of amyloid and tau, kind of the Bonnie and Clyde, of the brain, but I'm not sure which is who, male or female, doesn't matter. They drive each other on and they rob, not banks, but they rob the person of their abilities. So amyloid is probably there to protect the brain from infections. It repairs leaks in the blood-brain barrier. It promotes recovery from injury. It regulates synaptic function, as I mentioned. And in fact, a beta 42 and the so-called, that's the toxic form of amyloid, and the hyperphosphorylated tau, which is the tauopathy and tangles, they may be a tag team that actually start off pruning and development so that you're making some neurons make connections because that's appropriate for your environment. And as you grow up, you kind of, some pathways are encouraged and others are not. And that may be part of the pruning that helps you develop. They also protect from injury. And we don't fully understand that from infections, but also if you have a stroke or a head injury, you get a buildup of amyloid and tau around that stroke. It sort of walls it off, almost like an abscess. And so these are important proteins for health, but they also can become injurious. And there are other misfolded proteins, for example, something called synuclein. It's implicated in Parkinson's and Lewy body disease. And a new kid on the block we've only known about for maybe 10, 15 years called TDP43. And they regulate some aspects of RNA, that regulates some parts of RNA processing. And they can cause neurodegenerative diseases by themselves, but they're often comorbid with Alzheimer's disease. And this is why I want to show you this. Because the truth is that autopsy, and this has been replicated in other studies, not just this one, where they followed people, this is Chicago aging study, followed people to their death. They were often in their 80s and 90s and hundreds even. And here, just look at the probable Alzheimer's disease on the right. And the people that only had plaques and tangles were less than 10%. They had other degenerations, neurodegenerations. It could be synuclein, or it's like they think 40% may have synuclein, 40% may have this TDP43. But mostly they all had V. V is vascular, vasculopathies. And mostly people have just looked at the arterioles, the arteries, the arterioles. And as I'll argue, they've kind of forgotten the veins. And that's been something that is very relevant to the white matter disease. So in mild cognitive impairment, normal cognitive impairment, you see more vascular pathology, but the Alzheimer's disease pathology is only part of the story. So that's something very important to be aware of. And this was something that was not fully understood, when I got started in my career. And what we sort of did was we started phenobanking the common dementias in the real world. So these were patients who were coming to us in the clinic. We were able to get some funding to get going, and we were lucked out in getting a very good quantitative MRI protocol. We were able to do SPECT, which is a nuclear medicine technique that allows you to see blood flow, which is kind of like PET. We eventually could do genetics, psychology. We did all these things that allowed us to begin to understand, at least short of the molecular imaging, which has not been so available to us, how different proteins work together, and we follow people to autopsy. And I'm going to show you a little bit about our kind of research embedded in care, which allowed us to start doing this at one site. And now we have something called the Toronto Dementia Research Alliance, where we're binding together at the University of Toronto. We see 2,000 new patients a year. We're starting to develop standardized tools, standardized blood samples, and brain imaging, and then we can start to learn more about larger numbers in a faster way. Because the other thing is that Alzheimer's disease is not straightforward. Everybody thinks of just a short-term memory problem, and that's sort of the typical Alzheimer's disease. But it depends also on your age of onset, what your pattern is going to be. If you're younger, you might have something a little bit different, where it starts at the back of the brain, and you can have trouble with perception and language, but your memory is not so bad. You can have something called logopenic aphasia, which is you develop problems with word finding and language, not so much memory. You can have a frontal variant where you're kind of apathetic, but your memory isn't so bad. And then I think young and beyond, like the young people with the autosomal dominant age of onset, really they're dead by 50. So it's very aggressive because they're making amyloid from birth. So now with these different techniques, we can mark out the hippocampus. You've got to trace it right, and you can make measurements of the hippocampus and start to look at how it changes over time. This is called the cortical thinning signature map using FreeSurfer, where you can see the high points of vulnerable areas in the brain. That again, it's using kind of machine learning techniques. And we have many more options now for imaging. We can do amyloid PET since the early 2000s. The FDG PET's been around since the 1970s. The structural MRI, again, we can do the thickness, we can do various kinds of measures. And then we can also do microstructural integrity, looking more at the at the connections, the white matter that's connecting all the different parts of the brain. We can even do resting state, where you just look in a resting state when the person's ready to do something. How are these different centers connected? And they're different. There's kind of attention networks and resting state networks. All of this can be mapped out just with an MRI. So this model began to evolve, where there is this, as people were followed now with these different techniques, the PET imaging and so on, that there was a way that you accumulated these various abnormalities, with the amyloid being there to start before it actually caused much trouble. The tau was sort of coming along next, and then you began to get some shrinkage of different parts of the brain, especially the hippocampus. Then you start to get, memory affected and other areas of clinical, day-to-day functioning. So you go through from being cognitively normal to what's called mild cognitive impairment, where you have problems with your memory, but you're still very functional, to when you start to depend on other people, right? So what's interesting is we're getting now to the stage where we're going to be attacking the cognitively normal individuals with some antibody developments that may remove amyloid from the brain at a time when there's less damage and there may be more potential to prevent development of symptoms. But we're not there yet. It's still in the discovery phase. But there are actually right now there's a major trial going on worldwide doing just that. So that's the proteinopathy. Now let's just talk about the vasculopathy. I'm going to just fly through a bunch of pictures to get this across to you, because it's also very complicated and rich. And this just shows you all the different vascular cognitive disorders, that we have. There's kind of the vascular risk factors, which damage not just the body's vasculature, but also the brain vasculature. You can get large strokes. You can get small vessel, what they call lacunes. You can get hemorrhages. In fact, Alzheimer's disease, when amyloid goes along the vessels, can end up with a cerebral hemorrhage that can kill you. Alzheimer's disease is a cause of cerebral hemorrhage that can kill you. That is without any antibodies. But it's one of the things that the antibodies that are being used right now can accelerate. So people sometimes have died from the cerebral hemorrhage. We'll talk about that very quickly at the end. I'm going to talk a little bit about some strategic infarcts to show just how complicated this can be, but also cardiac arrest or being hypotensive. All of that can cause hyperperfusion, which can cause damage to cortical structures or circuits joining those structures. Can also affect something called cholinergic transmission, which I want to talk about later, because that's a part of the brain that involves focused attention. That's what the current treatments like donepezil and rivastigmine and galantamine, they're trying to maintain the cholinergic function by making it, synapses, making what you do produce, because it's reduced in Alzheimer's disease and also in Lewy body and Parkinson's disease, make it last longer so you get a bit more bang for your buck. That's basically how they work. This just shows you in the Sunnybrook dementia study, we've got now close to 1,600 cases, but we have people with Alzheimer's and small vessel disease, frontotemporal dementia, vascular cognitive impairment, Lewy body disease, and some normal controls. And this is where we've done some of our studies I'm going to talk to you about. So the first thing we realize is you can't just you have to pay attention to what's deep in the brain, not just the cortex. And so you have to kind of understand what this white stuff is. And I'm just showing you here a pipeline that we developed. It's kind of a personalized pipeline where we can get the gray matter. We can map out the white matter. We can look at strokes, but you have to trace them and you have to kind of subtract them out if you want to do the cortical thickness mapping, for example, or just get the gray matter amounts. And along these lines, we have been able to really be able to look at people with stroke and also Alzheimer's disease. And we've also been able to discover some of these perivascular spaces. I'm just going to show you those in a moment. The one point I would like to make, if you just send everything off to the free surfer pipeline and you don't look at what the outcome is, it can be kind of scary. And one of the reasons we started to realize we had to do our own more personalized approach is because it can over-erode and under-erode if you don't have the spinal fluid there to really give you the brain surface. So this shows you a free surfer case, a person with free surfer, where part of the frontal lobes are missing. And actually, sometimes even the anterior temporal lobe can be missing because there's some artifact from the mastoids underneath it. And so we thought, this has got to be customized as well. And so we do free surfer, but we do our head from brain because you have to do a T2 scan that allows you to see the cerebral spinal fluid around the brain in order to get the edge of the brain. And then you can be careful to correct for this type of thing. So there are multiple phases of small vessel disease. To the left down here would be a recent small lacoon. These are some of the white matter hyperintensities. I'm going to talk more about that. This is a cavitated lacoon. We actually get a hole, not all of them cavitate. These are the perivascular spaces. We're going to talk about those more. These are the microbleeds. All of these are ways that there's injury to the brain from vascular disease. And just recently at higher Tesla MRI scans where you get more resolution, you can see microinfarcts. These are like a millimeter. And this is often associated with atrial fibrillation. And they're usually in the distribution of the kind of middle cerebral artery territory, but it could be in any territory. And they're little tiny infarcts that you have to look for in order to see. But they are associated in memory clinics, where they've been looked for, with a greater chance of decline. So these are new kids on the block that require special attention. So let's talk about the inflow, the arteriolars, this kind of this kind of iceberg that I'm afraid they're melting all over the place. So it's not such a good analogy anymore. But here are the arterioles, and this is a very important slide for you to appreciate. So as you can see here, this is the front part of the brain, and these are the arterioles, and they're sending in, the surface arterioles, and they're sending in the arterioles down to drain into the venules that are around the ventricles here. You can see that there's quite a bit of territory where, in fact, they don't even come close to the venules. So the capillaries have, they've got to go through enemy territory to get to the venules. So this makes this part of the brain, kind of somewhat prone to be in oxidative stress, more so than other areas of the brain. Anyway, if you have a blockage of an arteriole, you'll have a little hole like this. This is a tiny one, probably not of too much consequence, but you have a lot of them. Since the cardiovascular health survey and the Rotterdam study, people started to look at these little holes, as well as the white matter disease, people started to realize that they were important. But let's look at we'll look at the medullary, we'll look at the veins later. I just wanted to compare notes because on the other side, you can see the veins, the venules, and they have transcerebral veins. They also have the veins that drain, and they've been sort of off the radar. Nobody seems to pay any attention to the veins. And that's why I think some of this white matter disease has seemed mysterious. So with the arteries, you can have hardening of the arteries. You get, blockage here, like in diabetes and hypertension. Hypertension is bad for the brain. You can get amyloid around the vessels. You can see here a nice vessel on the left, but this kind of messy looking vessel on the right, that's got amyloid around it. That is very friable. That can bleed easily. That's not going to deliver proper autoregulation, etc. So if you've got amyloid angiopathy in the brain, which is not uncommon in Alzheimer's disease, but it can even exist by itself in a hereditary form, then your arteries are going to be in trouble. But the one thing that people don't realize is you can have no vascular risk factors other than just age. And the age aspect of it is remodeling. And this was a beautiful little study done by a group in the US where Thor et al., where they looked at thick slices and they stained the arterioles in different decades. And you can see here's, a kind of arterial actual length when it's straight. And this is when it starts to remodel. So what they showed here is, this would be in your 50s, and maybe your early 60s, 70s, you get to your 80s, it varies, right? But you can see by here, over the right here, you've got no blood flow. So that's the same as having hardening of the arteries. You've got no blood flow, and it's just because of remodeling. You can also have a very tiny lesion. This is in the anterior, dorsal anterior nucleus of the thalamus. And I'm just showing it to rotate around because this is an example of a small strategic stroke where you're normal 1 minute. And when this happens, there's an artery occlusion, you are demented. And dementia means you have more than two areas of cognitive dysfunction. So what this does, this little strategic stroke, it sends radiations to the front part of the brain, which has to do with abstract thought, especially on the left. It sends it to Broca's area, which is where you find words and say, you can say things out loud, you can name words. And it also sends radiations to the fornix, which goes to the hippocampus. So it has to do with short-term memory. So you've got a person who has lost abstract thought, can't talk properly, and can't remember anything. And it's devastating because they never recover it from it. And I'm just showing you how a tiny strategic stroke can make your life change forever. And it's often not recognized. It's not recognized. It gets missed, by the way, for those of you who are familiar with FLAIR and various types of imaging, you might not even see it on a FLAIR. So I've had examples of people who were sent home because it didn't show up on a CT, they didn't get better, and then when we did an MRI properly, you could see that this was the cause. The size of those infarcts matter, and we showed that if it's over 55 cubic millimeters, then you definitely get, decline, especially on the left more than the right. But even just small white matter hyperintensities in the thalamus seem to be associated, with some changes in function. These are the cortical little tiny infarcts that I mentioned. You see how hard they are to see? You really have to be OCD to look for them. But there they are. You can see they're dark on T1. This is a 3D collection where it's like 1 millimeter thick. And then you can see them a little bit on a flare. But if you have a thick flare, you may not see them. Now, the perivascular spaces, it turns out they're very important because they seem to be the brain's lymphatic system. This was something that we could see because we were using something called proton density T2. And we could see that on proton density, you don't see hyperintensities that are raccoons, they're actual strokes. You see on T2 both the strokes and the perivascular spaces. But what we could do is we could distinguish a perivascular space from an infarct because just of this sequence. So it allowed us to begin to segment the perivascular spaces from infarcts because they can look the same. And also the perivascular spaces are a bit different around the basal ganglia and around the cortical regions. And so you have to kind of have different understanding of what's the basal ganglia is the old part of the brain, and the neocortex and all the white matter disease, the new part of the brain. And because we were able to do this, we could show perivascular volumes, perivascular space volumes were associated with disordered sleep after stroke, for example. Now we'll move on to the amyloid angiopathy. So here again, there are different regions. If it's deep, then they're more likely to be associated with hypertension, especially in the basal ganglia shown here. If you're seeing them out of the cortex, that means it's more likely to be related with amyloid angiopathy. Either way, you can, if sometimes if you get something called siderosis, where they gather around a whole gyrus, you can easily get a bleed from it. You don't want to hit your head. You don't want to hit a tree if you're skiing, because you'll definitely be in trouble. So that is showing outside is the perivascular, the conchophilic amyloid angiopathy. Here's a gentleman who was a lawyer, presented with confusion, and he bumped into desks at work, and the people thought he was drunk, and they took him into the emergency department. They did a CT scan, and he had a massive hemorrhage in the left parietal lobe. Affected his language, a whole bunch of different functions, and he never, was able to return to what he did before. A couple of weeks later, when we did something called gradient echo, which brings out iron in the brain, you can see that the iron in the hemorrhage is sort of healing and shrinking. You can see all the iron in it, but he also had little dots all over the place. So he didn't know it, but he had amyloid angiopathy. Amyloid meaning this amyloid beta 40, 42 along the vessels. It's mostly 40. Angiopathy means, pathology of vessels. So there were these little black spots, and often is a prelude to later development of Alzheimer's disease, which was his fate eventually. So about 5 years later, he went home, had to have somebody kind of with him, attendant. He was quite rich, so he had sort of a personal attendant, but eventually he started to develop memory problems and eventually died of Alzheimer's disease. This is the siderosis, where you have it along the vessels. And this can be very dangerous for bleeding. You hit your head and you might bleed. And we don't know what to do about that. We don't have a treatment for that right now. So it's a very dangerous thing. The other thing that you can have is this vasogenic edema, so-called, which started to be seen with some of the antibodies that didn't work so well, but they did sometimes stir up, almost like you're trying to get the amyloid out along the vessels a little too fast, and they overflow the exit, and you get this, what's called vasogenic edema, first noted in the early trials, and now one of the things we fear the most in the current antibody uses. So that just sort of shows you it was not there at the beginning, it appeared, and then it did disappear mostly, but not completely. And sometimes it will leave microbleeds in its wake because it allows red blood cells to escape into the brain, and that's where they become those little microbleeds. So this just shows over time what happened with this person. It went away completely. Now let's talk about the veins, because I think that's what's really been off everybody's radar. I'll try to get through this quickly to get to the end. I just want you to see what different amounts of white matter disease look like. So this would be 5 cubic centimeters, around 10 cubic centimeters here. mid 1990s, Charles DeCarli and some other people were observing, when you get up to that amount, it seems to start to have a little bit of cognitive effect, but below that, maybe not so much. Now you can see what like 25 cubic centimeters look like. And here's 50 cubic centimeters, which you've got like really massive periventricular, white matter change. Well, what is it? What is it? Well, some people distinguish between deep around the ventricles and 3D connection and just out to the periphery. They're called deep and periventricular white matter hyperintensities. This to kind of show you, if you do kind of an imaging segmentation, this is what the periventricular look like. This is what the deep look like. And some people speculate about which are more important for cognition. The clinical significance is that on many, in meta-analyses, that these are associated with greater risk of stroke, cognitive decline, dementia, and death. Increased risk of stroke is about three times. Dementia is about 2 times, and death is about 2 times. And also with decline in global cognitive performance, executive function, processing speed, all of this was kind of noticed, as well as poor outcome if you have a stroke and you have this white matter disease. It's also related in various ways to different aspects of cognition in some studies we looked at just recently, including semantic fluency, where you have to kind of give all the words you can think of that has to do with animals, all the animal words you can think of, for example. Global cognition, or the overall scales, like you have for the MOCA, or you have for ADAS-COG, those kinds of things. Language would be naming. Processing speed is often tested with something called the trails A and B, where you join dots on a page, and then you have to alternate between in order of number. Then you have to do alternating number and letter, which means you've got to kind of know what you just did and move to the next one. And it's actually a very sophisticated test used often for driving evaluation in the United States. Executive function would be, planning and organizing and carrying out activities. So all of these are affected by these white matter. I'm not going to bother with the mediation models, but it's showing that the white matter hyperintensities do actually affect the thickness of the cortex, and can indirectly, therefore be associated with things like naming and so on. There's been a lot of kind of questioning about what they do and what how are they connected to cognition. So use these mediation models now that have become more available, trying to delve into these relationships. The other thing that's very important in terms of strategic location is if the white matter hyperintensities actually affect the cholinergic pathways. These were mapped out by Meselim in 1998, just around the time that we were getting interested in all this. And so we created a little scale that would tell you if those areas were involved. And we found that if you involve those cholinergic pathways, you had more executive dysfunction and you had more problems with attention and concentration. And we showed this we became able to quantify it. We showed that people with a high burden in those areas had poorer memory function, actually smaller hippocampi even. We also showed in a stroke sample where we were doing trails A and B, and you could have either white matter disease, or could actually have a stroke affecting this area in particular right here, which is a very important pathway from the back to the front. If you had injury there, you were more likely to have problems with this alternating, like for task switching, such as in the trails B, where you have to go between numbers and letters, whereas just the size of the stroke or the amount of white matter disease might affect your speed. Even more interesting for those of you who might be neurologists is that we found that carotid artery tortuosity, and I don't know if you've come across this before, David, but it was an observation of one of our brilliant research radiologists. And he noticed that sometimes you get remodeling of the bifurcation of the carotid artery, and it can batter up against a very important part of the brain. It's called the substantia innominata, which is where the nucleus basalis of Maynard and the septal nuclei and so are. And the nucleus basalis of Maynard is where the cholinergic neurons are. So it's going to hurt the cholinergic neurons. And what we showed was that it was associated with atrophy of this area. It was actually probably pounding and injuring it. And independent of, it actually affected the basal ganglia forebrain volumes. And it was independent of hippocampal volume as well, that this seemed to be associated with some shrinkage, and that and in fact, we even showed, I'll just show you in a mediation model, because that was just showing it in a replication sample, that it seemed to be affecting the attention and working memory score, as well as the memory score. And this is a large vessel complication of just a chance that your termination of a major vessel going to the brain bothers a part of the brain that's very important for focused attention. So what exactly is this white matter disease? So here's an important point. I've got 10 minutes or so. Okay, here's an important point, and that is, I showed you those the veins and the arteries coming in at the front of the brain. And if you look here, there's a little angle here showing the distance from the cortex in toward the ventricles. And what it shows is that the fall off of the blood flow from the cortex to the ventricles, and it's particularly severe at the front because there's more distance. So it's kind of like you got a bad rap if you happen to be the cell that's involved in myelination, called the oligodendroglial cells. If you happen to be around the ventricle at the front, you are underperfused and you're in a poor neighborhood from the beginning. And interestingly, the very first white matter hyperintensities we start to see in even people in their 30s and 40s is around the frontal poles of the ventricles. At the back, when you go down a little further, there's a little bit of distance there too. But when you go back further, you're starting to get the temporal lobe area, the posterior temporal horn area. And that's where it was observed in people with autosomal dominant Alzheimer's disease. They start to show the white matter hyperintensities because they're probably trying to pump amyloid against gravity when they're lying on their heads, like most people sleep on their side or head or whatever, against gravity, and they're getting amyloid angiopathy that's injuring the vessels. So they show in the autosomal dominant patients that the first change they have compared to anything else in their brain, compared to their non-mutation carriers, brothers and sisters, is white matter hyperintensities around the posterior ventricles. So this is just to make you be more aware and realize that we can now, with imaging, we can actually see some of these veins. This is a special one that I like because it really shows that they're transcerebral veins. They go right across from the cortex down to the areas where they're draining into the deep sinuses. And you can see the brain is full of veins. And this is showing it on something called SWI, which is, it's also a sequence that allows you to see microbleeds. And actually, you see the veins so much that it's really hard sometimes to distinguish them from microbleeds. But that's just to introduce you to the deep medullary venular system, which is transporting, kind of blood in towards the venous sinuses. So these patchy periventricular white matter hyperintensities co-localize with these. And this is showing mapping in an individual, a 65-year-old that my colleague, Dr. Gao, actually mapped his entire deep medullary system in 3D. And it shows that these white matter hyperintensities co-localize with this area, with these venules. And in fact, other people have shown that it's around these regions that you get these white matter hyperintensities. And this is, again, just showing I've even got it in all dimensions to sort of give you a sense of it in 3D, showing that these are perivenular white matter hyperintensities. And in fact, what we think they are is we think they represent leakage and sort of stasis of the perivascular spaces. They're not like, they can't transport as well. What's happening is the veins are hardening, they're collagenosing, they're not getting their fair share, so they toughen up and they become fibrous. They're like they're draining, like they're pipes that are draining instead of live tissue. They can expand and shrink and do whatever they have to do if your fluid status is up or down. And so what happens is they start to leak. And this shows you around the posterior area here, you can sort of get this feeling of these linear structures. This is in T1 and this is in what's called PD2. And one of the reasons we know that it's probably vasogenic edema is because in highly myelinated tracts shown here, so this is something called the frontal occipital fasciculus. It's like a speed train from the back of the brain to the front, highly myelinated, and it just goes right through that vasogenic edema. Similar to the optic radiation, which is here, it's a very highly myelinated tract, and you can see that it goes through the vasogenic edema. And then also what's called the U fibers, which are connecting one gyri to the other. So that's the kind of thing you see when it's actually fluid that we're talking about. It's not demyelination. Now it can lead to demyelination, and that led us, this is just showing more of the same thing, and also to show that you get edema around the veins as they go out to the surface of the brain, the transcerebral veins, so you can see them going out. And you can even have little blebs shown here, like these little blebs, and they can get, over 1 year, we trace them, they can sometimes get bigger and smaller, maybe depending a bit on the fluid status, but some of them are becoming sort of more fixed, and you can even get venous infarcts in these so-called superficial areas. And then when we did the pathology follow-up in these individuals, which is the advantage we had of following people over many, many years to their autopsy, we found, lo and behold, that this white matter disease was associated with hardening of the veins. And you can see it by using a special stain called a trichrome stain. Most of the pathology studies that have been done worldwide do not do this. Only a few. Ours was the first, and then we persuaded another group to do it just so we could have a replication. But what it shows is there's some venular inclusions, little tiny ones, and almost everybody, and once you get into your 70s and 80s, they're all included. But there are also larger venules, and their walls thicken, they get stenotic, they occlude. There's also capillary loss in these areas. They're denuded of capillaries. So it's actually, it's a serious disease. And it's associated clinically with gait and balance problems, slow thinking, sort of being stubborn and sort of perseverative and so on, and having sort of executive dysfunction. It's called the subcortical ischemic vasculopathy. So what we showed was that the best predictor of the white matter hyperintensity volume was the stenosis of the large venules shown here. But there was a contribution from demyelination. It's just that it fell out of the equation. And we also showed, believe it or not, venous collagenoses in CADASIL, which is a hereditary disease. It causes disease of the smooth muscle and causes a great deal of problem. As well, you start to have strokes and can have seizures and have severe white matter disease by your 20s and 30s. We finally persuaded another group to replicate or to go and do trichrome stain, and they eventually had to shop it around to a lot of journals before they finally got it published in the annals. And this again shows the same thing in their sample, which is the Oregon aging study. So we have at least one replication. We can also see it in CADASIL shown here, where they have like nothing but white matter hyperintensity throughout all their brain pretty much. Believe it or not, there's even in MS, David might be interested in this, you get collagenoses of the venules in MS too. So what this has led to, I'm coming close to the end, is this idea that, there's amyloid, and I mean, there's amyloid building up and all these different kind of pathologies, but that vascular may be actually the very first one. And this was shown in a study of in the Alzheimer's disease neuroimaging collection, that the first abnormality that was found in people who eventually ended up having Alzheimer's disease actually was a reflection of their vascular disease markers. And this is an interesting idea that has kind of been put forward, I think, which is very interesting, which is where you have this kind of default mode, which is sort of in these areas here, is also at watersheds between the anterior cerebral circulation, the middle cerebral circulation, and the posterior circulation. So they're kind of watershed zones, which are zones of, kind of, unfortunately, they're very important zones for the default mode and for focused attention, but there are watershed zones that make you more vulnerable to changes, that have to do with your vascular health. So I'm just going to talk quickly about the move toward the fluid biomarkers. And then we know that we've known this from CSF that we can measure tau and amyloid in the brain, A beta 42. The big game changer is we can now do it in the blood. And it's using a technique called single immune molecular array. This chip is available in the United States for purchase. There's p-tau 181, neurofilament light, GFAP. GFAP is a measure of the astrocyte activation. Neurofilament light is a measure of kind of the damage to the axons. But these are now available to actually suggest a diagnosis of Alzheimer's disease because the phospho-tau goes up as the amyloid goes down. You can also measure amyloid itself, A beta 4042 in the plasma with mass spec, and CN precivity is available from WashU and is available. And just even recently, thanks to a Michael J. Fox project in a kind of longitudinal Parkinson's study, they have now found in CSF that they can measure synuclein. So we now have a measure of Parkinson's disease in the spinal fluid. And these all correlate very nicely with, the CSF measures, also with the brain measures and the molecular measures. I won't bother you with all that because there isn't time, because I just wanted to end up by saying we are in a new era. It's very exciting and it's also very daunting. This just shows you the Alzheimer's disease drug pipeline, in comparison to, let's say, stroke and other areas. It's a cancer. It's not so there's not so many options, but that's changing. And this is lecanemab, which is the drug trial with an antibody that's been conditionally, it's been approved, by a small subgroup of the FDA and likely to be approved in July. And it's the safest of the ones so far that have been developed. The aducanumab has been conditionally approved and it is available for purchase and it is being used in the United States. None of these are approved in Canada yet, but this just sort of shows you the sum of boxes is where you look. It's an interview which has to do with kind of clinical meaningfulness, showing that it did show beneficial effects over 18 months. It required 2 infusions a month. It showed that it got rid of amyloid very effectively, and it was, p-values were very significant. And then a cognitive score, a kind of a function, so they have these different function scales, all showed the right direction. So it's likely to be approved, and everybody's, wondering what it's going to cost and what it's going to mean. But here's some of the adverse events which are of importance, and that is that you did have adverse events in 7% that meant you had to stop, or a serious adverse event, which is 14%, but it wasn't so dramatically different than the placebo, and particularly deaths were the same in both, mostly from hemorrhage. And that's because you can have a hemorrhage just from Alzheimer's disease. 28K a year, 28,000 US a year. How are we going to handle that? This was the case for aducanumab. It was conditionally approved. Interestingly, one of the other antibodies failed, and their failure was taken to suggest that actually it does matter to remove amyloid from the brain. There's a prevention trial underway with lecanemab. It's called the AHEAD study, happening as we speak, and it's in people who are cognitively normal, but are at risk because they have evidence of a p-tau elevation, which means they've got amyloid deposition, and they're at risk of declining. So the idea is to prevent them from becoming symptomatic, not to become a mild cognitive impairment, which means you've got a memory problem, but you're still functioning, or your early AD, which is you're functioning in a lot of areas, but you're still not quite right. So that's underway as we speak. But here was something pretty scary. In the same New England Journal, when they published the lecanemab results, they showed a person who had been in the trial, and I think she was on open label, and she had a left middle cerebral artery stroke and came in on time, so they gave her TPA, and she died almost on the table. And when they did the autopsy, they found that she had something that looked like some sort of vasculitis. So they didn't have all the data about how long she'd been on the drug, which had been on open label for a while. So I thought it was sort of an irony, because advances in one major, devastating disease, i.e. stroke, meets up with another devastating cause of disability and killed the person, right? So I think it does mean you have to be careful. We certainly know you have to be careful about anticoagulants and those sorts of things that can make you bleed, but this was the cause of death. And it was quite sobering, and it was published in the same journal, the same issue. Then finally, the top line results of the donanemab study needs to be mentioned, because it is the most efficient remover of amyloid yet. And they were able to show very significant reductions, not just beneficial effects in the clinical ability or clinical function, but also they were clearing plaque by 52% by 1 year, which means it opens up a whole new possibility that you would treat until you have no signal. That is, it's below the radar. It's still there and it's still happening, but then you follow the blood test, and if it starts to go up again, then you treat. So it'd be like chemotherapy. And it wouldn't be that you would have to be on these drugs, forever and ever. So they also showed much less decline. They were less likely to progress to the next stage. This is all just kind of coming out now, and Lilly will probably be going for approval very soon. This just shows the p-values are very impressive. So stay tuned, because I think there may really be some effective treatments coming. I'll just mention that focused ultrasound may be another option to get amyloid, get molecules into the brain more efficiently. And there's even some interest in using focused ultrasound in combination with the antibodies, and maybe using the focused ultrasound to open up the blood-brain barrier in a strategic way more gently to get rid of some amyloid, and then maybe use the disease modifying therapies in more detail. So a new era. I think we should also mention that there's anti-tau antibodies also being looked at. There are repurposing of drugs, semaglutide in particular. You mentioned about diabetes. It's all the rage for losing weight, but it's also being tried by Novo Nordisk in Alzheimer's disease. They think it's attacking neuroinflammation. There might be gut microbiome, but still important for us all. Healthy lifestyle, vascular risk factor management, including treating sleep apnea, it's fundamentally important. Back to the heart-brain connection, and back to this recent study that came out showing the population attributable risk of that are modifiable factors for dementia. Some of them are, early life, less education. They're accounting for different percentages. So less education. Everybody should be educated to give their brain a better chance. But the other thing that came out, because we're getting better at sort of smoking, smoking and diabetes, we're getting a little better. They're sort of certainly on the curve, but not accounting for as much, because we've been doing a better job, at least in developed countries. And so what's come out recently is hearing loss, which is really important. And we don't really know what's going on there, except you're probably understimulating a very important part of the brain in Alzheimer's disease, which is the temporal lobes. So that's come out as something, again, that could be remediable, and we have to try to make sure we treat. Traumatic brain injury, hypertension, alcohol use, as you know now, is starting to be under the gun because of its association with cancer. But these are all, many of these are lifestyle choices. Some of these are treatable conditions. Some of them are not. Social isolation and social engagement. The social engagement is actually beneficial. So, and air pollution, God help us, right now in Canada. But that still means there's a lot of risk that we don't understand, that may have to do with genes and other things. But I just thought this would be a good way to stop, to just remind you all that we all have a duty to try to do our best to have a vascular healthy life. And I just sort of summarizing all this and pointing out that we are moving towards now precision medicine, but that includes embedded in physical activity, sleep, diet, social engagement. And I think we are in a transformative transformational period. I don't know about you guys in the United States, but I can tell you in Canada, we're so unprepared that it's actually pretty scary, because I think it's going to be neurologists who mostly have to be involved in these treatment trials. In fact, we need teams. We need social work, nursing. We need teams in order to do a proper job here. But it is going to be, it's going to be on the horizon. So just want to acknowledge many sources of support. Some members of my team, Sandra Black, which right now you can't see anything in Toronto. Down at the end there is the CN Tower. It's all smog. But this is Sandra Black Health Sciences Center, where I work, and these are some of my support systems, some websites. And this is just for David to show the venous collagenoses in MS, where it's more an adaptive immunity. It's a T cell injury, but it still injures the veins. Thank you.
Well, Sandra, listen, I felt like I was back in fellowship again. I was a trainee listening to you. So it's a great flashback, by the way. So listen, thank you so much
My pleasure.
for being
Thank
here
you.
today for that presentation.
Thank you for the opportunity. Thanks very much.